My name is Dr. V.C. Patel, retired professor, Anand Agriculture University and Parul University, Anand, Gujarat. So the today topic is secondary plant nutrients, calcium, magnesium, sulfur, their functions, deficiency symptoms in plants. So secondary plant nutrient, calcium, magnesium, and sulfur. So secondary plant nutrient are as significant as primary nutrients NPK in plants, but they are needed in smaller quantity, whereas NPK requirement is greater in larger quantity. They are necessary for the augmentation levels of roots and cell. So here you can see that magnesium deficiency, calcium. So importance of calcium and magnesium ratio in plant growth and crop production. While calcium is a part of every plant cell, it keeps cell wall upright and increases fruit set and quality. Magnesium is a component of the chlorophyll molecule, therefore essential for photosynthesis. The mobilization of phosphorus can play a vital role in transporting and forming sugar and starches through the plant. Magnesium and calcium are not only important because of the role they play during plant growth, but also because of the interaction with each other, which is a key consideration when making soil management decisions. For example, high calcium levels can reduce the ability of magnesium, while greater magnesium level can induce calcium deficiency. So, balances of calcium and magnesium in soil is required. So, it will be seen that the ratio within a wide limits had no effect. The presence of sufficient quantity of calcium and magnesium in all soil is essential for the profitable production of crop. If the soil calcium and magnesium level are adequate and soil pH is acceptable, variation in the calcium to magnesium ratio between 2 and 8 have been shown to have no influence on crop yield. So when the calcium magnesium ratio ranges from 2 to 8, but there will be no any difference in crop yield. Calcium and magnesium influence on soil physical characteristics. Stable aggregate, favorable structure, and good tilth are characteristics of healthy soil. Calcium and magnesium promote aggregation. The calcium ion is smaller than magnesium. Hence, it is more efficient in binding soil particles and has more fluctuating power, the ability to clump and form small masses, than magnesium. Conversely, magnesium could lower soil structure stability. Calcium in the form of gypsum, calcium sulfate may be used to amend soil with structural problems due to sodium. The calcium component of gypsum replaces sodium in soil and makes sodium clay soil. So the sodium is subsequently removed from the rooting zone with either natural rainfall or irrigation. So when we reclaim the problematic soil, then leaching is required. Then how is the calcium magnesium ratio calculated? One examples, calcium and magnesium are determined by laboratory analysis. The ratio is calculated using the molecular basis, electrical charge basis. For example, if there is 4.88 milliculon calcium per 100 gram soil and 1.72 milligram per 100 gram, then the calcium magnesium ratio is 2.8:1. This value are typical for Iowa soils. Soil calcium magnesium ratio naturally are above 1:1. Many research trials since have not shown an influence of calcium magnesium ratio on crop production. An example is work by McLean and colleagues in 1983, in which ratios were manipulated by application of calcite lime, magnesium oxide, and magnesium sulfate, and yield response was measured. The results indicate that the highest yielding treatment and lowest yielding treatment both occur with calcium-magnesium ratio that span the same ranges, thus indicating the calcium-magnesium ratio was not the reason for major yield differences. So there will be no significant influence in calcium-magnesium ratio for crop production. So conclusion from the research where the results strongly suggested that the maximum crop yield emphasis should be placed on providing sufficient but no excessive level of each basic cation rather than attempting to attain a favorable basic cation saturation ratio, which evidently does not exist. Various greenhouse and field trials indicate that crop productivity is not influenced by ranges from less than 1:1 to more than 25:1 calcium magnesium ratio outside of what is normally measured in soil. Plants also play a role in calcium and magnesium uptake and exclude excess calcium or magnesium at the root surface. So in summary, the calcium-magnesium ratio concept is unproven and should not be used as a basis for fertilization or liming practices. Having sufficient level of calcium and magnesium is proper method of evaluation. So when there is a deficiency of the calcium and magnesium, then we have to apply. Rather than trying to manipulate ratio, there is no need required to manipulate the calcium-magnesium ratio for increasing the crop production. Then calcium, function of calcium, aid in the movement of carbohydrates in plant, essential healthy cell wall and root structure. Deficiency symptoms of calcium. Calcium is not mobile and is not translocated in the plant. So symptoms first appear on the younger leaves and leaf tips. The growing tip of root and leaf turn brown and die. Calcium deficiency is not often observed in plant because secondary effects of high acidity resulting from soil calcium deficiency usually limit growth. Precluding expression of calcium deficiency symptoms. Without adequate calcium, which in the form of calcium pectate is needed to form rigid cell wall, newly emerging leaves may stick together at the margin, which cause tearing as the leaves expand and unfurl. This may also cause the stem structure to be weakened. In some crops, younger leaves may be up and crinkle with the terminal bud deteriorating. Buds and blooms fall prematurely in some crops. So calcium deficiency usually are found only on very acidic soil. So calcium deficiency we are observing in acidic soil. They can be corrected by liming with calcium carbonate. Calcium is rarely deficient when soil pH is adequate. So when soil pH is adequate, 6.5 to 7.5, then there will be no deficiency of the calcium, but it will be found deficiency in acidic soil. So deficiency of sulfur, magnesium and calcium in tomato plant. So you can say here, a sulfur deficiency in tomato, magnesium deficiency in tomato plant and calcium deficiency in tomato plant. So leaf curl in tomato plant. Calcium deficiency in brinjal. Calcium deficiency, corn leaves fail to unfold. So you can see the leaves are unfolded. Calcium deficiency in maize or corn. Then calcium deficiency in cotton. So the leaves are called unfold in cotton. Similarly, we will find in groundnut also calcium deficiency. Then calcium deficiency in mango crops, horticulture crops. Then function of magnesium. So an ingredient of chlorophyll aid in translocation of starch with the plant, essential for formation of oil and fats. Deficient symptoms of magnesium. Because of magnesium is a mobile element and part of chlorophyll molecule, the deficient symptom of interval chlorophyll first appear in older leaves. Leaf tissues between the veins may be yellowish, bronze, or reddish, while the leaf veins remain green. Cone leaves appear yellow, striped with green veins, while crops such as potato, tomato, soybean, and cabbage show orange-yellow color with green veins. In severe cases, symptoms may appear on younger leaves and cause premature leaf drop. Symptoms occur most frequently in acid soil and soil receiving high amount of potassium fertilizer. So when we are applying high level of potassium, then there will be deficiency of the magnesium. And magnesium also deficiency we are observing in acid soil. So magnesium deficiency on acidic soil can be corrected by liming with dolomite lime, that is calcium, magnesium carbonate, magnesium carbonate. If the soil pH is sufficient, magnesium can be supplied with Epsom salt, magnesium sulfate, or potassium magnesium sulfate. So extractable magnesium soil test categories and suggested fertilizer rate recommendation. So in low category, when extractable or soil test magnesium less than 60 ppm or less than 0.5 millicurie per 100 gram soil, then the recommendation pound per magnesium per acre, or in the bracket I have given kg per hectare. So 10 to 100 or 11.21 to 112.1 kg per hectare. When medium category, 60 to 300 ppm extractable or soil test magnesium, or 0.5 to 2.5 milligram per 100 gram. So the recommendation of magnesium 0 to 60 pound per acre or 0 to 67.3 kg per hectare magnesium. Then high greater than 300 ppm or greater than 2.5 millicurie per 100 gram soil magnesium, then there is no need to apply magnesium in soil. Then intermediate chlorosis caused by magnesium deficiency in corn or maize. You can see here interveinal chlorosis. So vein remain green, but other portion become chlorotic. So here you can see clear-cut differences of the magnesium deficiency in maize or corn. Then cowpea magnesium deficiency, then red gram magnesium deficiency, then magnesium deficiency in brinjal, it is also known as eggplant. Then magnesium deficiency in potatoes, you can see here typical characteristics of the magnesium deficiency. Then magnesium deficiency in mango, magnesium deficiency in mango, you can see the leaf of the banana. Then function of sulfur, aid in the formation of oil and parts of protein molecules. So sulfur required for oilseed crop, pulse crop, and legume crops. So deficient symptoms of sulfur, younger leaves are chlorotic with evenly lighting color veins in some plant, as for example citrus. The older leaves may show symptoms first. However, deficiency is not commonly found in most plants. Growth rate is retarded and maturity delayed. When there is deficiency of the sulfur, then growth is retarded and maturity delayed. Plant stems are stiff, thin, and woody. Symptoms may be similar to nitrogen deficiency and are most often found in sandy soil that are low in organic matter and receive moderate to heavy rainfall. So we are found sulfur deficiency more in sandy soil because in sandy soil there will be low organic matter and the sulfate has been leached in sandy soil. Then sulfur deficiency symptoms in emerging corn, maize crop, you can see here. Then corn sulfur deficiency symptoms. Then rice sulfur deficiency symptoms, you can see yellowing belt due to the deficiency of the sulfur in rice crop. Then here you can see the sulfur deficient wheat plant left as light green leaves you can see here, and stunted plant as compared to normal wheat plant where sulfur is sufficient, means healthy plant. So on the right side we can see the healthy plant having sufficient sulfur, whereas on left side sulfur deficient plant are stunted and yellowish color. Then green gram sulfur deficiency, green gram also known as mung. Then brinjal sulfur deficiency, then sulfur deficiency in onion crop, then groundnut sulfur deficiency, you can see the yellowing of the leaves, then sulfur deficiency in cotton crop, and you know that at present the farmers are growing bt cotton in larger area and they are getting more remuneration. Then sulfur deficiency symptoms in tissue culture banana plant, then sulfur deficiency symptoms in banana plant. You can see typical symptoms of sulfur deficiency in banana plant. Then sulfur deficiency in mango plant. Mango is a horticulture plant. So control measure for sulfur deficiency in plant. So when we are carry out this soil analysis, then when there is a lesser than 10 ppm sulfur, then it is a low category. So we should have to apply sulfur at the rate of 20 to 30 kg per hectare. In different of sulfur sources, gypsum, then elemental sulfur, then sulfur coated urea, and single super phosphate. Then when the range is 10 to 20 ppm sulfur, then it comes in medium category. So apply sulfur at the rate of 10 kg per hectare sulfur in different form of sulfur sources, gypsum, then elemental sulfur, sulfur coated urea, and single super phosphate. But among the different sources, gypsum is a cheaper source and it also works as a soil amendment. It improves the soil fertility. Then in high category, when the greater than 20 ppm sulfur, then it is a high. So sulfur is sufficient in soil, so no need to apply sulfur fertilizer in soil. So you know that we can observe the calcium, magnesium, and sulfur deficiency on plant. But for confirmation, we should have to carry out soil as well as plant analysis, and then we have to apply the different sources of the sulfurs. You know that at present, I am working as a chief soil health specialist at Sevama Agri Clinic and Agri Laboratory, Bhavnagar, and we are carried out the soil, water, and plant analysis. And on the basis of this soil, plant, and water analysis, I am giving advice to the farmers on soil test value. And so far we have carried out 5,000 soil samples, and the farmers have the opinion that they are getting the response by applying our recommendation of the fertilizer or manure, and they are getting the crop production increase of about 20 to 35% yield as compared to the control. So soil analysis is very important. So please contact to our Sevama Agri Clinic and Agri Laboratory, Bhavnagar, Gujarat, and our website is sevama.in. And if you have any problem, then please contact. My name is Dr. P.C. Patel. My email is patelpc1212@gmail.com. Again, my name is Dr. P.C. Patel. My email is patelpc1212@gmail.com. Thank you. If you have liked my video, YouTube, then please subscribe. Thank you very much.